Genetic engineering is defined as isolating the DNA of one organism and placing it into another for replication purposes. This process is used in multiple facets of science and technology, such as medicine, agriculture, pharmaceuticals, biofuels, and much more. In these fields, genetic engineering or gene therapy answers questions pertaining to sustainable agriculture, genetic diseases, and renewable sources of energy. However, ethical and political concerns arise from the ambiguity surrounding this newly discovered method. Despite the obstacles that genetic engineering may face, with further intensive research, this field will make rapid advances in the next century. In the last 30 years, genetic engineering has been refined to a series of steps that starts with the building blocks of life in one organism, DNA, to a newly created superorganism. The process begins with a short segment of DNA called a gene that codes for a specific protein. Proteins provide the basis of observable traits in an organism. Once scientists discover a superior trait in an individual, they are able to isolate the gene and determine the genetic code for the protein. After isolation, the small chunk of DNA is inserted into a plasmid, which contains all of the necessary components for replication. When there are enough replicated segments of the gene, the plasmid can be inserted into the target cell for further replication and protein synthesis. No matter which facet of genetic engineering is being discussed, the process is the same for all fields of study.
From a biofuel perspective, researchers are working on creating renewable energy sources that are safer for the environment while also sustaining the economy of America. This is done by breaking down compounds from fuel in order to view their protein structures that provides the basis of energy. From there, taking these proteins and manipulating the structure of corn, algae, and other cellulose-based plant molecules enable the newly synthesized proteins to be inserted. Once the proteins are coated, replication occurs and the engineered algae or corn will display enhanced properties of energy stability. Genetic engineering in agriculture began commercially in 1996. By 2010, genetically modified crops occupied 148 million hectares in 29 countries across the world, with most production in the United States and Asia. Currently in the US, 75% of processed foods available in stores contain genetically modified organisms. Genetically modified crops grown for food are designed with the intention of increasing production, changing nutritional content, and creating herbicide and insect-resistant plants. While there is much controversy about the long-term effects of changing these genetic properties of crops on the environment and related organisms, thus far there have been minimal adverse effects on the environment reported by researchers. Overall, the economic effects of using genetically modified crops in the United States are positive. Increased production leads to reduced need for importation and greater possibilities for development of crops for other uses such as biofuels.
Genetic engineering in pharmaceuticals, commonly referred to as farming, involves the use of genetically modified organisms to produce pharmaceutical products and mainly includes the mass production of insulin, human growth hormones, vaccines, and many other drugs. The process of developing and generating a profitable farmed product can take years, and the process is considered experimental. But this process has been very beneficial in being able to produce many types of drugs and hormones for medical use. One of the major pharmaceutical products as a result of genetic engineering is insulin. It was actually one of the first genetically engineered products. Insulin is extremely essential in the treatment of diabetes, a chronic disease in which there are high levels of sugar in the blood. Previously, insulin extracted from pigs was used, which have dissimilar amino acid sequences as compared to humans, so it was common for humans to experience immune responses. But with the help of genetic engineering, insulin can be mass-produced, absolutely identical to human insulin, not causing immune reactions. Another important pharmaceutical product, human growth hormone, or HGH, can now be extracted from genetically manufactured bacteria, and it can be used to treat those suffering from dwarfism. The main goal of genetic engineering in pharmaceuticals is to produce and allow access to these medicines in more efficient ways.
Gene therapy refers to the use of modified DNA to replace mutated or disease-causing genes in unhealthy cells. The genes are inserted into a vector that will allow the DNA to be integrated into the host cell's DNA without being rejected. The host cell will then be able to produce normally functioning proteins. Though there is not much currently known about the side effects associated with gene therapy, this type of medical treatment could potentially provide a cure for several diseases, such as Alzheimer's disease or certain types of cancer.
Started over. I started over.
Wait, time out. Let me just make sure.
I hit this little button.
Okay, good. It saved the
I
total.
know what I'm doing.
That's all I just want to make sure it saved the total. You have to talk for 2 minutes. 1 minute and 59 seconds.
The political ramifications of genetic engineering are widespread, cross-disciplinary, and fundamental in nature. Questions involving everything from basic human rights, moral considerations of the unborn, and property protection to safety and disclosure arise across the various disciplines and industries which apply genetic engineering technologies. Delta Force One considers two major aspects of political issues with genetic engineering. The first are human issues. Diagnosis using genetic engineering technologies allow medical professionals to diagnose genetic conditions with genetic engineering. Now, the issue here is that employers and insurance companies may want to know the results of these kinds of tests. Employers, of course, want to know if their employees will be affected by some disease or condition in the future, and insurance companies may want to deny coverage or increase rates for those with pre-existing conditions. Delta Force One is opposed to compulsory disclosure in this area. We believe that people have a right to their privacy, and that unless the existence of some disease or disorder diagnosed by genetic engineering has a direct impact on the rights of some other individual or individuals, they shouldn't have to disclose it. The other human issue is prenatal diagnosis. This is similar in that medical professionals can diagnose genetic disorders in the unborn. There are several issues here. First, treating the unborn here. We are in favor of treating the unborn when it's safe. We don't really see any problem with that. However, abortive measures when, for example, an unborn baby. The second area of genetic engineering political issues that Delta Force One considers are the agricultural issues. Agriculture companies develop genetically engineered crops called genetically modified crops, or GMCs, that have certain desirable traits like resistance to disease or better resistance to environmental conditions or easier reproduction. And so they develop certain seeds that are genetically engineered and then sell them. The issue is whether their intellectual property rights should be enforced here. Whether farmers can continue to use their seed without paying for them again, whether they can sell them to other people. And Delta Force One is in favor of protecting the rights of intellectual property holders in this area. However, we do recognize that it is difficult to enforce. It's difficult to regulate the distribution of a self-producing, self-replicating commodity. The other issue with agriculture is safety. Are GMCs safe? This is another controversial issue. We believe that with significant government testing, we can rule that they are safe, and when they are safe, we are in favor of GMCs, but we believe that they must be tested in order to be proven safe. The other issue here is compulsory labeling, whether companies should label those genetically engineered crops as such. This we think is unnecessary. If the crops and the produce, the food, has been proven as safe, we see no reason to mandate compulsory labeling of genetically modified crops as such.